Hello everyone and welcome to the first EANO Youngsters Journal Club. I am Maximilian Mair from the Medical University of Vienna, and I'm really excited about our first session and also the high number of registrations and participants today. So these are my conflicts of interest. Before we dive into today's paper, I'd just like to give a short introduction on this brand new format. So the Journal Club is planned as a 1-hour webinar where we discuss new papers and cutting-edge research in our field. Interdisciplinarity is in the DNA of neuro-oncology, and that's also reflected by our Journal Club, as we will discuss papers spanning from basic science to clinical research and covering also the diverse spectrum of specialties in our field. So we're looking forward to lively discussions, and the Journal Club also aims to increase the participation of youngsters within our society and therefore provide a platform for networking. So who are we? The EANO Youngsters Journal Club Working Party is a diverse group of youngsters of various countries and fields in neuro-oncology. So in fact, it's the first working party of EANO. So the first group of EANO members who are working on a project outside of the established committees we have in our society. So if you want to actively participate in the organization of this new format, you're very welcome. Just drop me an email or feel free to contact me also on social media. Before continuing, I just wanted to draw your attention to other EANO activities specifically tailored to youngsters. First, we have the EANO mentorship program, where you can work on a project with an experienced expert in the field. Second, the EANO seminars, where specific topics are addressed in a 2-day format this year in March in Warsaw. And lastly, the annual meeting, which takes place in Glasgow this year in October. So don't miss to submit your abstracts and don't miss the youngster sessions we are planning right now. So today's session is a best of 2023 session where we'll discuss the results of the INDIGO trial, which was published in the New England Journal of Medicine this year. So you can actively participate by sending us your questions and your comments. All you need is the Q&A feature in Zoom. So just click on Q&A, put in the questions and click on send. So we will pick them up and discuss them later on. So in the end, I'd like to thank you for your participation. Please also share your feedback. This is a QR code where you get directly to an evaluation form for this first session, but you will also get an email with an evaluation link after this session. So with that, I'm at the end of my short introduction presentation. I just stopped sharing my screen now. And with that, I'd like to introduce my co-chairs and speakers of this session. So first of all, Tanja Müller from the University of Frankfurt, who is also a youngster and will discuss the paper with us. Then our two senior and experienced experts, Matthias Preusser from the Medical University of Vienna, and also Martin van den Bent from Erasmus Medical Center in Rotterdam in the Netherlands. Very happy to have you here. Thank you for your time, especially also you, Martin, as you were directly involved in the INDIGO trial. And then, last but not least, Angelo Dipasquale, who will lead us through the paper and also then build the basis for a lively discussion later on. So, Angelo and also INDIGO, the stage is yours.
Thank you, Max. Thank you for your introduction. So yes, let's start this first EANO Journal Club session with the paper Vorasidenib in IDH1 and 2 mutant low-grade gliomas, published. I cannot go on. Okay, quite slow. Published on the New England Journal of Medicine in 2023 by Mellinghoff and colleagues. Here my disclosure. And yes, we know that as for now, with the WHO 2021 classification of brain tumors, also diffuse glioma center finally the molecular era, and now 2 molecular markers, the IDH mutation and the 1p90q co-deletion, are able to distinguish gliomas in 3 different entities with very different prognosis and treatment approach. The glioblastoma IDH wild type, the astrocytoma IDH mutant, and the oligodendroglioma IDH mutant and 1p90q co-deleted. We know that the IDH mutation is the biological marks of this subset of lower grade gliomas. And in general, the IDH mutation leads to several biological modification in the tumor cell, inducing impaired cellular differentiation, alteration in metabolism, and also epigenetic changes, including genome-wide methylation of DNA. And today in clinical practice, patients with IDH mutant glioma usually receive a maximal safe resection followed by a wait-and-see strategy or a sequential radiochemotherapy according to several risk factor of progression, in particular the grading, the age, and also the extent of resection. We know that radiation and chemotherapy are able to improve prognosis of our patients, but our patients live very long with their disease, 10 to 20 years of median overall survival. And we also know that radiation and chemotherapy may affect quality of life. For example, for radiation inducing in the long term some neurocognitive dysfunction, in terms we can see of executive dysfunction, of working memory impairment, and also of attention impairment. And we also know that chemotherapy is able in some way to impair the fertility on time. And we can see from this experience how a lot of patients still have interest in having children after the diagnosis of an IDH mutant glioma. So looking for new strategy to defer the use of radiation and chemotherapy in this population is a very urgent need. In 15 years of research about IDH mutation in gliomas, we finally arrived to a phase 1 trial about vorasidenib. Vorasidenib is a small molecule that is able to inhibit both IDH1 and 2 mutation, is able to have some activity on IDH mutant gliomas, in particular for non-enhancing gliomas. We can see, for example, a reduction in the tumor growth rate over time with the use of vorasidenib in these phase 1 trials. And also we know that vorasidenib is able to have a good blood-brain barrier penetration. So all these early phases data, with all these early phases data, we move to the Indigo phase 3 trials. The Indigo trials included patients with a diagnosis of an IDH mutant grade 2 residual or recurrent oligodendroglioma or astrocytoma, according to the WHO 2016 classification. Patients should have received surgery only for their disease in 1 to 5 years before the enrollment, and should have a measurable disease according to the EANO low-grade gliomas criteria, and also should have no need of immediate radiation or chemotherapy as for physician judgment. These patients were randomized 1-to-1 to receive the vorasidenib 40 milligrams daily or placebo. And also there was a stratification for the 1p90q status and also the tumor size at the enrollment. Also, very important, at progression, a crossover to the vorasidenib was also permitted. Here are some words about the statistical design. The primary endpoint of the trial was progression-free survival, so time from the randomization to the first tumor progression. And the key secondary endpoint was the time to next anti-cancer intervention. 3 pre-specified analyses were planned, and in the second analysis in September 2022, there was a confirmation of signal of efficacy on vorasidenib. Here are some data about the state of the enrollment at the second interim analysis. We see 77 centers involved, a median follow-up for patients of around 14 months, and also around 331 patients included in the trial. A very important point, in March 2023, the Independent Data Monitoring Committee recommended the unblinding of the trial after the early demonstration of efficacy in the second interim analysis. In this case, most of patients crossed from placebo to vorasidenib. Here we are having a look at patients' characteristic. We can see a median age of 40 years for both vorasidenib and placebo arm, preserved clinical condition in general with a very good Karnofsky performance status, a median time from the surgery to the randomization of about 2 to 2.5 years, and a 50-50 ratio for the inclusion of both oligodendroglioma and astrocytoma patients. Another important point, most of these patients have a longest diameter of the tumor of 2 centimeters or more. And in particular, the median age of 40 years and also the tumor size of 2 centimeters or more probably describe the patients with a high risk of progressive disease according to the most recent guidelines. Here we can see the primary endpoint, the progression-free survival. So vorasidenib improved progression-free survival over placebo with 27.7 months of median PFS compared to 11.1 months of median PFS. And someone can ask if there is any difference between the progression-free survival and the time to next intervention. The answer is yes, because in clinical practice, the RANO progressive disease does not mean every time it is time for a next intervention. This is a case from my institution where over a RANO progressive disease, the disease of these patients with oligodendroglioma was considered quite minimal. So the decision was to go on with a wait-and-see strategy. And yes, the key secondary endpoint, the time to next anti-cancer intervention, was also reached with a median time to next cancer intervention not reached for vorasidenib compared to 17.8 months for the placebo arm. Here, there is a subgroup analysis. We know that subgroups analysis in general are quite explorative and hypothesis generating, but we can probably see some signal of activity from vorasidenib in patients with high risk of disease. So in patients with 40 years of age or more, in patients receiving more than one surgery, so the recurrent tumors, and also in patients with a longest diameter of 2 centimeters or more. Here, this is probably another important point for our patients. Vorasidenib was a well-tolerated drug. We see, for example, a grade 3 or more adverse events in around 20% of patients, with the increase in the alanine aminotransferase as the most common grade 3 adverse events in around 9 to 10% of patients. And another important point is that the discontinuation of vorasidenib due to a treatment-related adverse events was seen only in 3.6% of patients. So I think now discussing indigo is a very complex task, and I think the very big question in the beginning is how to stop this chain reaction of questions the trial is generating in the neuro-oncology community. So I think it's time in this part of the presentation to start our discussion. Tanja, it's up to you.
Thank you very much, Angelo. This was a very comprehensive summary of one of the research papers in the neuro-oncology field of last year, I guess. I guess most of you are already familiar with this publication and with the study. I would like to give the audience the chance to type in their questions into the chat. I think you, Angelo, have prepared some points that we could discuss. So we were thinking about to start the discussion.
Yes, just to break the ice, maybe we can start with a very big question, probably which is the ideal candidate to vorasidenib now in clinical practice, where we are falling in the middle between the low-risk patients and the high-risk patients according to most recent guidelines. And I don't know if Matthias and Martin want to join the discussion.
Martin, we cannot hear you, but I know you have a strong opinion on this high risk versus low risk classification.
Yes.
But, you want me to say something about that?
Yes.
Okay, I'll be happy to share some thoughts. So routinely when we're talking about animals, we're not considering the mammoths anymore. They died out many years ago. So the entire thing about the high risk and low risk of low-grade gliomas is based on the era prior to the IDH discovery. So all the thoughts about age, performance status, size of the tumor are really based on observations that are old-fashioned, are not consistent anymore with what we currently are seeing. So if you look today now at the at attempts to see how much prognostic information there is in histological grade of IDH mutated tumors, that's actually very little. The same thing holds for that tumor remnant, less or more than 2 centimeters. That doesn't mean a lot. And if you look at the age distribution, you will notice that most of the patients, because of their age, the median age was 40, and 80 or 90%, I don't know the exact figure anymore, had a tumor that was bigger than 2 centimeters in size. So if you look at the old guidelines, 90% of the patients had a high-risk tumor, and they're just slow-growing tumors. So we should forget about these considerations, and we have to reinvent the ways we are looking into the prognostic classification of these tumors. And if you look today more carefully in papers that looked in real depth to prognostic factors, and look at the volume of the tumors, and especially the volume after surgery, you will see that the volume after surgery is probably the most important prognostic factors in the life of an IDH, of a patient with an IDH mutated tumor. Probably more important than grade, definitely more important than age. And even in a grade 3 tumor, you have ample time to look at the patient, to look at the patient characteristics, and try to find out what's the best next step for that patient.
Also, in regard of this point, what about molecular profiling? Could this be a tool to define which patient belongs more to the to the low, to a high, or intermediate risk group, or which patient could be treated with vorasidenib?
Yeah, there are some thoughts here as well. We have some very nice papers over the past few years that looked into this. Of course, we now have homozygous deletion of CDKN2A, which is now automatically qualifying the tumor for a grade 4. And outside that, there are a few other consistent observations like CDK4 amplification, PDGFR amplification, other hits in the RB pathway that are associated with a poor outcome. And I think the next WHO will take some of those factors into consideration. For oligodendrogliomas, it's more complicated. There have been a few papers that looked into this in more detail, but it's less clear. Homozygous deletion of CDKN2A is also found to be of prognostic relevance in anaplastic oligodendroglioma, but it is always found in anaplastic oligodendroglioma, so the WHO did not use this as an additional feature. So yes, there are some observations on prognosis and outcome, but what we don't know is to what extent these molecular alterations predict the outcome to vorasidenib. We just don't know that, and we have to find it out. I think that's one of the next missions that we have as a field, is trying to figure out if there are any of those alterations that are associated with a poor outcome that really predict the outcome to vorasidenib.
Oh, I think this question, which is the ideal candidate to vorasidenib, it looks simple when you look at it, but in fact it's a very complicated one, because even if you take a very restrictive point of view and say, okay, it's those patients who fulfill the entry criteria to indigo, I think even that is pretty unclear because some of the inclusion criteria are in fact ill-defined, even tumor grade, right? So you would think that a grade 2 tumor is a grade 2 tumor, but this is not the case. If you have several neuropathologists looking into the tumor grading of a certain case, you will find that some of them will call it a grade 3 and others a grade 2 tumor. Grade 4 is a little bit different because due to the fact that molecular parameters are involved here, it gets a little bit more clear. But the distinction between grade 2 and grade 3 is really very shaky. Even if you look into the WHO book, and Angelo, you had the book on one of your slides, and you read the chapter on how to do grading, it's very astonishing how ill-defined the criteria are, be it mitotic frequency or signs of anaplasia and so on. This is not very well defined. So I think even saying restricted to grade 2s is more complicated in real world than it seems. And I wonder what the implementation of vorasidenib into clinical practice will do to neuropathologists. It might be that they feel some kind of pressure to diagnose, in case of doubt, a grade 2 tumor rather than a grade 3 tumor, if that means that the patient has a higher likelihood of receiving the drug. Another parameter that is difficult in clinical practice, I think, is the contrast enhancement. So in indigo, patients without contrast enhancement were included. But what does that mean, really? We all know these cases where there's a little bit of patchy contrast enhancement here and there. Does it mean that's no contrast enhancement? And how will neuroradiologists report it in their findings? So I'm not aware of a EANO classification on patchy contrast enhancement or something like that. Yeah, and so even if you say you want to give it to patients who fulfill the indigo criteria, that's not so easy. And then the question is whether that's a good strategy, or whether we would feel comfortable enough with the data to extrapolate that to tumors that might not be 100% fulfilling those inclusion criteria. Grade 3 tumors with a little bit of contrast enhancement, tumors within the first year after surgery, tumors after 5 years of surgery. How do we feel about those? And then looking into the future, it will be very interesting to see how regulatory authorities actually word the label for the drug. So indeed, I agree, many things to discuss.
Yeah. Then I have maybe one related question. So if these are patients with recurrent or autoresidual disease, why was placebo the control arm and not radiochemotherapy? Maybe Martin, you can comment on that because you were involved in the trial.
So the trial was set up in a patient population in which it was felt reasonable not to treat with radiation therapy and chemotherapy. And I would think if you do that, you compare two completely different approaches. So here, vorasidenib was really positioned to see if you can, in that early phase of the disease, if you can postpone, if you can mitigate the clinical outcome of these patients, and that was proven right. And I think the use of a placebo, of course, it had a lot of discussions in the with the regulatory agencies when the trial design was proposed to them, and that's one of the reasons that it was quite restricted. So it was grade 2 and unenhancing. So a patient population in which everybody agrees that it is reasonable to sit back and wait. You're not harming the patient if the tumor progresses to some extent. But that then comes back into all the discussion. So what was investigated was a drug in grade 2, non-enhancing disease. But by the biology of the disease, why would it be limited to grade 2? Why would it be limited to absolutely non-enhancing tumors? This was the safety aspect of the trial.
Yeah. Definitely important thing to consider. There is another question of one participant that is maybe related to that. Felix wrote, aren't most of the tumors in indigo comparable to those enrolled in RTOG 9802, so the trial comparing radiotherapy to radiotherapy plus PCV in grade 2 gliomas. If so, the progression-free survival in Indigo for the experimental arm was 2.3 years, in RTOG with RT and PCV more than 12 years. Could Professor Preusser and Professor van den Ven please comment on this?
Yeah, I think the purpose of vorasidenib is not the comparison with radiation therapy and chemotherapy. It is how can we postpone radiation therapy and chemotherapy. And most of my clinical scientific work has been related to investigating the addition of chemotherapy to radiation therapy. And the issue that we see is that in those patients that survive long, after 6, 7, 8, 9 years, they start to show difficulties with maintaining their normal quality of life. They are dropping out of work. They have a difficulty with noisy environments. They have all the problems with the cognition, memory, because of the long-term side effects of radiation therapy and chemotherapy. So yes, we have a very effective strategy, but it comes with a price to our patients. And this is what we hope to be able to delay with vorasidenib if we can postpone radiation therapy. And I don't think we know yet the amount of time that we get by using vorasidenib. The follow-up period of the trial is still so short, but I think that is where the value lies, that you can postpone radiation therapy for a prolonged period of time with a treatment that's of quite low toxicity.
Thanks. Another question from the audience. It's from Mircea Tesileanu. I'm sorry if I didn't spell this in the correct way. So bevacizumab worked well for progression-free survival, but did nothing for the overall survival. So do we believe that vorasidenib is going to have an effect on overall survival? And what would it clinically mean if it would only be beneficial for the progression-free survival?
I'll be happy to take that one. Mircea is a PhD student of mine. Mircea, I first want to remind you of the fact that more people have difficulty in pronunciation your name.
Sorry.
Yeah, I said it wrong many, many times, and he keeps being irritated by that. So this trial obviously does not give an answer to the overall survival. And for a trial for overall survival, you would need a follow-up period of like 14 to 18 years. And it was decided very early on that that will not be a realistic scenario. So that was not tried. And we don't know that. We need to start following these patients. And we need to find the real world data of these patients. We need to set up cohorts of patients and follow them and see in the end whether we are impacting that survival. But it will really take young neuro-oncologists to fulfill that mission, as we will get a readout in 20 years from now.
Yes, but and the question was, even if there is no OS benefit, is it worth it? Is it still a valuable treatment? I think yes, because it might help to delay radiochemotherapy and thereby kind of buy time. And at the same time, it's a well tolerable treatment. And we haven't seen this data here, but at SNOW it was also shown that it decreases the growth rate of the tumors. And we will see what the effects on quality of life and so on are on the long term. But I do think it would be a worthwhile treatment with these PFS data, even in the absence of an OS signal.
We have then 2 questions regarding the molecular longitudinal development and evolution of gliomas. So first question is, what are your thoughts on the reports of cases when a tumor is IDH mutant at initial diagnosis, but later due to chromosome deletion or other mechanisms IDH is lost? Or in cases when hypermutation occurs and IDH mutation is not the main oncogenic driver? So how may vorasidenib have an influence on that? Or
yeah, the clear answer is that we don't know. It probably is the explanation that those trials on late-stage disease with hypermutated recurrences or enhancing disease failed to show any efficacy of the IDH inhibitors. That's what you see in almost all the IDH inhibitors. Those studies that carefully examined low grade of unenhancing tumors versus enhancing tumors saw efficacy in enhancing tumors and not in the unenhancing tumors and not in the enhancing tumors. And one of the reasons of the failures of some of the compounds could be that because they were investigated in phase 1 study, the late stage diseases, they didn't see efficacy. One of the important questions that we have to ask as a field, and we have to answer as a field, is what will happen with those tumors over time when patients start to progress under vorasidenib? Have molecular features of the tumor changed? Will they remain similar responsive to radiation therapy and chemotherapy? That hypermutated recurrence, is it associated with temozolomide? If we postpone temozolomide, will that be delayed? All these questions require real-world data, good follow-up of cohorts of patients that receive treatment, and most importantly, trying to get surgical specimen of patients that progress after vorasidenib and analyze those cases. So I don't think the question is answered with this one trial with a relatively short follow-up. We really need to come up with large cohorts of patients that are followed over time and answer these type of questions consistently. So we need to work together on this.
Yes, and there's also the question of combination therapies. For example, IDH is known to be immunosuppressive, so would combination of IDH inhibitors with some kind of immunotherapy, be it immune checkpoint inhibition or vaccination or something like that, be rational and useful?
We had another question regarding the differentiation, but I think this is kind of addressed. So Thomas Kashta was asking whether it's possible to estimate from the data available so far whether there's a higher risk of differentiation to higher grade gliomas. Yes. Yeah, but I think this was kind of addressed, if you agree. Some more technical questions regarding the study. So do you know maybe, Angelo, how frequent the MRI imaging were done during the trial?
Yes, we can go here again to the randomization. Here, every 3 months for 3 years, and then every 6 months.
And maybe just coming back to the big topic of the molecular background. Is there any translational data available from poor responders to vorasidenib coming from Indigo? Question of Pasquale.
So as you will see that there are only a handful of poor responders that underwent second surgery. We just have a few relapses, and of those few relapses, only a handful have underwent a second line of treatment, and there are only a few cases, there's less than 5, that underwent surgery from which a specimen is available. So we don't know, and the company is trying to collect these cases, and they are following up all the institutions and ask them to submit tissue samples if they are available. But you can imagine that if a patient is showing progressive disease under vorasidenib, there is no second surgery, and they go right away to radiation therapy and chemotherapy, and we don't have these tissue samples. Again, this is going to be a very important question for the real world data study, the projects that we need to conduct as a neuro-oncology field.
Thank you very much for this answer. I have another rather provocative question from the audience. So following up on Martin's answer before, but if we use IDH inhibitors as a first line to delay radiotherapy, we have preclinical evidence that reduction of 2-hydroxyglutarate through IDH inhibition leads to radioresistance. So there's obviously a paper of Dipasquale, 2015 in Cancer Cells. And we do not know if radiotherapy is still effective as a salvage treatment then. Shouldn't we be much more careful?
Yeah, there were actually similar papers on chemotherapy as well, because some of the resistance mechanisms against chemotherapy are also depending on ketoglutarate. So if you refill the amount of ketoglutarate, that might also hamper the chemotherapy sensitivity. The problem is that not all the model systems that have been used have resulted in the same consistent signals. And the model systems are not IDH mutated low-grade glioma cells. They have always been highly dividing cells in which IDH mutation was introduced. And even the few that have been looking at cells that you can culture from an IDH mutated tumor, that's always the ones with the CDKN2A mutation. So they're not the typical low-grade IDH mutated cells. So yes, I think that is a concern. That's a concern, and I look forward to the American trial that's going to be done, in which vorasidenib will be combined with radiation therapy and chemotherapy, because I think that is a key question. Again, it speaks to us setting up real-world programs, because we need to answer that question in due time as well. If the only way to answer that question is to start collecting systematically all the patients, the data from all the patients that are being treated with vorasidenib, and see what happens with them afterwards. If we don't do that, we will not be able to answer these questions. At this point in time, there are just too few patients undergoing the next line of treatment that we can answer that in a reasonable way. For that, you need real-world data.
Thanks a lot.
Yeah.
Tanja, sorry.
No, yeah, I also would like to think this is, we see it all the time, circles around that we need more data in the future, yeah, to see how's the long-term outcome. Another question, also, but more technically, not really technically. How many patients had to discontinue vorasidenib due to deranged liver function tests? Do you know, or do you have it in your slides?
I have only this slide about the safety profile, but we don't have across data of discontinuation and increase. But I think the discontinuation was quite low for the adverse events. So
yeah.
So discontinuation was in the 4% range.
Okay.
And there were 3 of the well over 100 patients, so it was less than 2%, I think, that had severe hepatic toxicity, but they all recovered after discontinuation after a few weeks. So yes, it is a drug that needs careful monitoring.
And maybe another question to both of you, Matthias and Martin. So any thoughts on the prospect of PET imaging or MRI spectroscopy for the measurement of 2-hydroxyglutarate levels in evaluating the tumor burden and response to IDH inhibitors?
So I personally think that's a very interesting question. We don't have data on that. Patients in Indigo didn't undergo PET imaging or advanced MR imaging, but I think metabolic imaging is an interesting field to explore, because as we discussed, we don't know which patients to select really, which patients have a higher or lower likelihood of responding. So we need better biomarkers. And for example, by amino acid PET imaging, you can delineate tumors better than by MRI imaging when it comes to contrast enhancement, right? So amino acid PET uptake is typically larger than the areas of contrast enhancement. There are tumors that light up in amino acid PET imaging, but not with contrast enhancement and so on. So it's not the same information you are looking at. So that might be useful for patient selection and also for follow-up. But there are also other tracers that might be of interest in the future for PET imaging. There are IDH tracers, so you can directly visualize IDH. These are not available for widespread clinical use at the moment, but are under development. And MR spectroscopy and other MR-based techniques are certainly also very valuable to get more information on the tumors and thereby use this information as patient selection criteria or for follow-up. So I think we as a community should really invest research into that and try to collect information on that. So when vorasidenib becomes available in clinical practice, it might happen that some groups amongst us start scanning patients with different modalities at baseline and during treatment and see whether and how that can help clinical practice.
Thanks. So at the moment, we do not have questions from the audience, so please feel free to put in your questions again. Maybe Angelo,
Yes,
you
maybe we
could
can
proceed
go
with some
on
further.
with, yes, with some other questions, because yes, we say that we did not see, so it's quite difficult to distinguish between a grade 2 and grade 3 gliomas. And I was wondering, for example, if methylation profile is able to refine grading for the adjuvant gliomas and then help us in selecting patients for vorasidenib, patients for radiation and chemotherapy upfront, or maybe just patients for a wait-and-see strategy. What's your opinion on that?
So what you're showing now is actually the work of Mircea, my PhD student, who did very nice work, and it was further taken forward in a part of the GLASS study in which paired samples were investigated. And it makes me believe that with methylation arrays and looking at methylation of specific CPC sites, we might be able to come up with a good prognostic classifier of low-grade gliomas. Pim French, working in the laboratory over here, has some very nice results now. And I think this may help us to understand better which patients have a poor outcome and have a better outcome. That still doesn't tell us, however, if these profiles are predictive for the outcome to vorasidenib. Things can be prognostic, but without giving us information on whether it helps to identify patients yes or no responding. So we are sort of all assuming that it's the patient with a favorable outcome that may respond well to vorasidenib, but we haven't investigated that. One of the downsides of having very strict inclusion criteria is that it doesn't allow you to talk about generalizability of the findings to a more broad population. And again, I come back to what I said before, we need real world data on a more broad population, trying to figure out which factors are associated with a favorable outcome. The issue will be that outside a clinical trial, it will be very difficult to come up with predictive markers, because for that to assess predictive markers, you actually need a control group that was treated differently. And in the absence of such trials, it will be difficult to come up with an answer to that. But still, if we are able to find factors that are associated with almost no response to vorasidenib, predictably almost no response to vorasidenib, of course, that will be very meaningful.
Yeah, it would be maybe also interesting if we have some neuropathologists listening to us, that they can share their thoughts about the grading and what this could mean for the future. Maybe just you can share your next slide, Angelo.
Yeah. Yes, we talked somehow about this, so the difference in magnitude of progression-free survival between vorasidenib and radiation plus chemotherapy. But yes, probably the answer is there are a lot of molecular pathways that are able to lead to the development of resistance to vorasidenib in clinical practice. But yes, we need more molecular data, I guess. And probably this is another interesting point. So we imagine vorasidenib in other clinical scenarios, I don't know, as all-comers drug, so every patient should receive vorasidenib and then radiation chemotherapy at progression. Or maybe we may think of combining radiation and chemotherapy to vorasidenib at the same time. Or maybe there is a role for vorasidenib at progressive disease to radiation and chemotherapy.
Yeah, well, these are all questions that will need to be addressed in clinical trials, and actually within the ERTC we're exactly thinking about these questions and have ideas on all of these scenarios. For example, the combination with radiation therapy and chemotherapy. What does it mean? So we're thinking at the moment about maintenance strategies. So in terms of clinical trial design, of whether it would be helpful to give vorasidenib after completion of radiation and chemotherapy, rather than combining it upfront, right? But I mean, that could also be a strategy, of course. And the big question will be also, what about patients that have progressed after radiochemotherapy? And there are many patients out there that have this problem or will have this problem in the next months and years. So can you put those patients on vorasidenib? At the moment, no, because there are no data about this. So I think that's one area where we definitely need the clinical trial very soon.
Are there any further questions from the audience? So please put in your questions. So if I'm right, crossing over was allowed in the trial. So was crossover allowed in case of progression by RANO, or was it allowed as next treatment? So because these are the two major endpoints that were analyzed.
Yeah, so it speaks to the limitation of that endpoint, next time to next intervention. Like you said, I think like 35% of patients in the placebo arm crossed over to the active drug at the time of progression. Patients could be deblinded. It had to be progression confirmed by the independent radiology review committee. But at that point in time, the patients that were in the placebo arm were offered treatment in the active arm. That means two things. One is that overall survival is not an endpoint that you can ever assess in this trial, if you allow that progression-free survival is the best that you can do. But I also think it somehow polluted the time to next intervention trial, because it was kind of easy. Patients that showed some progression were offered that opportunity, of course, which was attractive to patients. And if you see that the case also shown by Angelo, you see a little bit of growth. Okay, you stop the patient from continuing the study drug, but it doesn't mean you have to go right away to radiation therapy and chemotherapy. So I don't think that this study shows the value of that endpoint time to next intervention. And I think the data shows basically the progression-free survival. What it does show, however, is that those patients that were on Vora, more than 85% were still not undergoing any other treatment after 2 years. But again, you need more follow-up in order to be able to make to draw some conclusions on that. But I think that part of the study is not the most robust.
We have then another question from the audience. Are there any strong data confirming the efficacy of vorasidenib in pediatric patients as inspiration for future trials?
So the study started at the age of 15, I think. And
12.
12, okay, even 12. So most of the IDH mutated patients are above the age of 12. It's only rarely that you find a patient with an IDH mutated tumor below the age of 12. It does happen, but in astrocytoma, but it's very rare. The amount of patients under that young age are very limited. I would not see a biological reason why they would respond differently.
Then we have another question also discussing this issue of progression by RANO. So Francesco asks, MRI volume analysis has a higher sensitivity to assess small and early changes in tumor response during vorasidenib, whereas a longer period of observation might be needed to assess the consistent response according to the standard RANO criteria. Do you think that new criteria based on volume MRI are needed to design new trials in IDH mutant gliomas?
So that's a discussion that has been ongoing for many years. And what it actually needs is that somebody shows in the data set that meeting volume is better than meeting two-dimensional. It has been said many times that, for instance, for glioblastoma, it would be better to measure volume. But if you look at the people that did trials on that and tried to make a comparison between 2D and volumetric assessment, whether that would correlate better to overall survival, the answer is no. It doesn't correlate better in overall survival in glioblastoma. So that speaks to keeping it simple. The classical way of meeting solid cancers was two-dimensional. So the old WHO criteria were two-dimensional, and the McDonald's criteria and the RANO criteria for low-grade gliomas were derived from those two WHO criteria. The RECIST criteria are one-dimensional. And why were they introduced? Well, people investigated it and felt and showed that it correlated as good as two-dimensional and was more easy to do. And therefore, the solid oncology field moved towards RECIST is one-dimensional. Now, what we have on information about RANO, 2D, and volumetrics is basically limited to the paper of Ben Alexander and his group, which showed that the inter-observer assessment, inter-observer variation is smaller in volumetrics than in 2D. But they haven't shown that it correlates better with outcome. But here you have the problem again that if you want to correlate with outcome, you have to wait for those many years before you get to that point. What they did show is that there was a change in progression-free survival using volumetrics. But in the absence of a control arm, a control study, you actually don't know what you're looking at. So I think at this point in time, it's probably safe to say volumetrics allows you a better quantification. But if it correlates better with outcome, which would be the one thing that you would like to see in order to change and to make everybody start using volumetrics, which is a more complicated tool, and not all the hospitals have that in their portfolio. I think that's my expectation is that we are going there, but it will take time before the technical solutions are there to allow that real time, very fast, that clinicians can use it. If you look at the classical low-grade tumor, the area of whiteness and all the scarring that is around that, it's still quite complicated to make a distinction between what's really tumor and what's scar tissue, radiation therapy effects, etc. It's an ongoing discussion. My expectation is the same. In due time, it will become volumetric, but it will take a couple of years.
I think we have one last question. We are already nearing the end of our journal club, but one last question. Just one additional thought, given the potential use of vorasidenib to delay neurotoxicity from radiotherapy. Many of that notion is based on older trials with old radiotherapy techniques. Newer trials, for example, EORTC 22033, showed no difference in health-related quality of life or mini-mental status after 3 years, comparing temozolomide versus radiotherapy. So then again, radiotherapy is not always that bad, especially with new radiotherapy techniques, IMRT, VMAT, protons, and one should be careful to restrict its use without a clear head-to-head comparison, especially with the lack of efficacy of radiotherapy after IDH inhibitor use. So was more a comment than a question.
Valid point, maybe, yeah.
Yeah, it's a valid point. Maybe Martin, your take on that?
Yeah, so just to show the limitation of the EORTC study, a mental status examination is not a valid tool to assess neurocognitive functioning of patients. It's just way too simple for that. And the health-related quality of life assurance in all the academic trials is very limited in the period of follow-up. So in the 22033, I think we reported on 1 or 2 years, and then after the compliance to the measures becomes so low that you cannot make any conclusion anymore. So yes, we have health-related quality of life data, but only for a very small period of time. And it's my impression that even with more advanced radiation therapy techniques, still patients over time develop more issues. So we are doing a better job, I think, with advanced radiation therapy techniques, but I'm not sure that we really solved that issue.
Thank you. And I think we take the last question as there's only one left, and I guess it's a quick one. So over the past decades, various IDH inhibitors have been developed and investigated. Some examples are noted here. And is Vorasidenib the final step? And what improvements would you like to see? A question from Jan Michael Werner. Do you have some comments, or someone has a comment on that?
Well, we have a first drug. It will be interesting to see if other drugs are better. We don't know yet the duration of progression-free survival. We have a first estimate, but we don't know if that's the end of it, if there's more possible, if there are other compounds, mechanisms of resistance, can they be overcome with other IDH inhibitors? I think there are tons of questions. We just have now a first proof of principle that generates so many other questions, also with other compounds. Could 2 compounds be better than 1 compound? Just as a question.
Okay, so I think it's already time to wrap up. So thanks you all for your participation. Thanks, Angelo, for your very great and comprehensive presentation. Thanks to Matthias and Martin for your time and your expertise in this journal club. Thanks, Tanja, as co-chair. So that was our first session of the journal club. I will just again share the QR code for the evaluation. Just a moment. So here we are. So please share your feedback. Please fill out the online evaluation form. An email will follow with the link to the evaluation form. So thanks you all. Thanks to the participants, and see you at the next journal club session.